Hello, everyone. My name is Santosh Kulkarni from Syngene International. So thank you for joining this call today, and thank you for attending this seminar. So what I'm going to talk today is about the RNA-targeted small molecule discovery approaches. And just to give you a background about the research area, as well as what we have been doing. We at Syngene have been supporting small molecule research primarily in the protein-based areas, and slowly we are entering into RNA-based targets. And I just wanted to share with you some of our own experience, as well as our expertise in this area, as well as what's new in the realms of RNA-based drug discovery. So before I get into that, let's start this talk. And anyone who is entering new into the RNA-based drug discovery has to understand what is RNA. I mean, this slide probably shows you a basics of biochemistry, but as we all know that the RNA is a central part of protein translation machinery, which actually transmits the encoded information from a nuclear genome to a cytoplasm. So as you can see here, the DNA, which is actually the encodes for the information, which then transcripts to get the RNA, which then translates into produce the proteins. And we all know this is a central dogma in molecular biology. With the completion of the Human Genome Project, what it was realized is that only 3% of the human genome actually encodes for the protein. And we all know that the RNAs in this play a major role. And so far we have been understood that the RNAs encompass mRNAs, which encode for the protein, and the ribosomal RNAs, which are involved in translating this information. Well, there is not to it. I think RNAs play even multiple roles. And these actually are called as coding roles as well as non-coding roles. And in humans, there are majority of the RNAs actually have a non-coding role. So what you call as a non-coding RNAs. And some of the early studies with the functionally as well as in the different disease states actually has led to believe that some of these non-coding RNAs are actually involved in cellular processes, as well as not just in regulating the transcription, but also in terms of translation and impacting the epigenetic markers as well as the cellular signaling pathways. So that clearly sort of opens up a new era in biology, which actually opens up new targets which can modulate with small molecules. Now, essentially, some of these RNAs or non-coding RNAs are sometimes dysregulated in diseases like cancer, as well as neurodegenerative, as well as neuromuscular disorders. And sometimes these are also differentially expressed in disease as well as normal states, again, showing a selective modulation in the disease states could allow a small molecule approach for finding a therapeutic agent. So clearly, I think the era of RNA field has opened up a new targets, or something what we call as undruggable targets, and that's something which is very exciting in the field today in terms of targeting the RNAs. So it's not that the RNAs have not been targeted. I mean, as you can see, RNAs have been targeted with small molecules. It's just that we didn't know about it much. Especially in the bacterial and the viral therapeutic agents, I think we all know that these actually, some of these molecules actually do target the RNA or the RNA proteins. Antibiotics like aminoglycosides, tetracyclines, linezolide, as well as chlorphenicol actually targets the bacterial ribosome. And believe me, these compounds were not discovered rationally, in the sense like these were actually discovered using the traditional approaches of screening the fermentation broths or the compounds to identify the targets. It was fortuitous that these molecules did bind to the targets such as ribosomes, as these are in abundance, clearly showing the importance of these molecules or chemical matter in the RNA field. More recently, I think Merck has shown the discovery of a molecule called ribosyl, which actually inhibits the flavin mononucleotide riboswitch. And this is something also known to disrupt the translation downstream mRNA. So clearly showing another approach actually to find a small molecule in the bacterial RNAs as a targets. And there have been some work which has gone in the HIV, especially targeting the TAR as well as the RRE elements in terms of targeting. So clearly, there is a precedent of using a small molecule to target the RNA or RNA binding proteins as a successful therapeutic agents. So the interest in RNA-based small molecules for human disease has actually increased with the advent of or the success with some of the molecules which recently got approved, and especially in the area of RNA splicing. So RNA splicing is a process where the precursor mRNA is actually transformed into mRNA or a fully functional mRNA by removing the introns and joining the exons. So as you can see in this figure, the process of transforming this actually leads to a functional mRNA, which then encodes for the protein. And there are several processes in between which can be modulated with a small molecule to actually impact the disease. And there have been success in this, and as you can see here, there are approved drug called in the 2020 called risdiplam discovered at Roche and PTC, which was approved for spinal muscular atrophy, which actually does by enhancing the splicing of the SMN2 protein transcript. So actually essentially increasing the SMN protein, which is essential for maintaining the muscle structure. So in the similar approach, Novartis actually has moved a molecule called Brenner-Plum, which is now in phase 3 trials, again, functioning by the same mechanism. Although, as you can imagine, from these small molecule drug-like molecules, the advent of RNA-based drug discovery has taken into a new leap, and clearly showing the use of the small molecule approach towards modulating the activity of RNAs. Again, some of the recent studies have shown that these small molecules not only just bind to the same target, but bind in different locations, again showing the complexity of the RNA-based targets in the modulating the small molecules. Recently, the HT Biomedicine has disclosed this macrocyclic molecule, which is entered into phase 1 clinical trial for leukemia, again functioning by impacting the splicing factor B3b subunit 1. So clearly, I think the non-coding RNAs or the RNA splicing is a very important target or the arena to look for modulating with the small molecules. So here on this particular slide, actually, you can see the diversity of mammalian non-coding RNAs. Again, it's a huge list. Still, and the function is still growing, and we are learning more about this. As these are arranged with respect to increasing size of the RNAs. And as you can see, the microRNAs are involved in gene expression through specific RNA slices silencing. And these have been actually known to modulate in different diseases. And there have been success actually at least in the RNA space, where people are able to find small molecules which actually modulate the activity of microRNAs. One other the non-coding RNA which has gained a lot of interest is actually long non-coding RNA, especially like MALAT1 as well as hot air, which are become like a very attractive targets actually too, because these actually are expressed in a specific tissues, and also in the regulatory functions in the of these have been altered in disease states, clearly providing opportunity to find a small molecule which can be then progressed into clinic. So as we learn more about these non-coding RNAs, I'm sure there'll be a lot new interest in actually going after these targets to identify novel therapies. Now, certainly, I think one can come from a protein-based drug discovery to actually use those similar technologies actually to RNA-based. Well, there are certain differences, and some of these actually can be seen is because of the simplicity of the RNA structure, because of the 4 bases, but which is there. So basically, the RNA is actually a very simplistic molecule if you look at it from a 2-dimensional structure, but it can be sometimes complex because of the folding or the structural folds that it goes into. And this structural complexity can be seen from the recently elucidated structures of RNAs, subsequently ribosomes, which shows the possible structural complexity of the folded RNA. So when this particular RNA folds into a different structure, actually that can give into a complex fold, and the ligand binding pockets can be identified in this. Now, because these RNA folds are thermodynamically more stable, also can lead into different ensembles of structures, again, adding more complexity to the understanding of the structural folds in the RNA. Single strand RNAs have been known to fold into different secondary structures, like by the base pairings, like structures like hairpin or internal bulge or loops as well as bulges. So clearly, these are actually the different structures which actually can form a different binding pockets, and that can be then targeted with the small molecules. These again can fold into higher order structures because of the highly dynamic nature of the RNA where it exists. And these can be impacted with the interacting proteins as well as salt concentration as well as in the small molecules. Again, opening up numerous ways how one can target RNA or RNA folds with the small molecules. Can also lead to some disease states, for example, in the case of neurodegeneration, like especially the tau neurodegeneration as well as frontotemporal dementia. One could see a differential folding of the RNAs can lead to different diseases. So again, the RNAs can also exist in higher order structures like tertiary structures or the quaternary structures, something what you call as a triple helix junctions, pseudonodes, as well as G-quadruplex, which actually can form a different binding pockets, and that then can be utilized for targeting with the small molecules. So clearly, I think the structural complexity of RNA is still need to be elucidated, and that provides enough opportunity to actually target with a small molecule to identify new medicines. Now, what are the different ways how one can think of targeting RNA-based targets? Now, coming from a protein-based approach, I think can be sometimes good, but sometimes can be lead to unsuccessful efforts. But what I see, or what we see as three different approaches one can go after based on the target or based on the different non-coding RNAs. Now, these have been evolved based on the success as well as based on the experience with whatever the targets and functions what we know today about the RNAs. Now, the first one is, of course, is the phenotypic screening of compound libraries, which is actually a standard approach in the industry today to actually go after the RNA targets, wherein this has been successfully applied in the example I just presented to you in the RNA splicing programs, where a mini-gene construct with a luciferase reporter was used to actually screen compound collections, and that led to identification of good hits. This approach is very valuable for RNA targets where you have tertiary structures as well as RNA folds or interaction partners, which actually have a defined binding pockets, which can actually target it with the known compound libraries what we have for proteins, which are generally hydrophobic, as well as the interactions can be similarly exploited. Now, STS of drug-like compounds has advantages, as we all know that this can give you good hits, and that can lead to a good starting points in a medicinal chemistry optimization plan. Now, the further confirmation of these bindings with the binding site analysis or the structure-based approaches, as well as affinity tagging, has been successfully in actually taking these molecules towards optimization. Now, we have certain examples, or we have some experience in that, and which I think was quite successful in actually taking some of the early hits into lead out programs using this approach. Now, when you look at the more simplistic RNAs, which are like these hairpins, bulges, as well as internal loops, those are more like requires newer techniques, or sort of what I call as RNA-centric approaches, which requires a different approach for finding a chemical matter for these targets. Now, these structures possess unique binding motifs, highly charged states, because as you know, the RNAs have a charged backbone with a very polar interaction, so would require a different approach in terms of looking at the small molecules. Now, there are known scaffolds and fragments, which we call as the RNA binders, something what we can imagine as a privileged structures, which can be used as a starting point. Then using the chemoinformatics tools as well as the library design approaches, one can actually design libraries or the molecules which will be starting off from these privileged structures of RNAs to these find a new starting points. Now, these compounds can be enriched with more heterocyclic binders as well as an activity-guided approach to actually enhance the compound collection. And this is the area where we'll see a lot of progress coming from a lot of industries in terms of publications as well as disclosure of new RNA binding motifs, which will help the community. Now, as a de novo approach, I think there is also a very fantastic work being done in some of the academic as well as industrial labs, especially from Matthew Disney and his group, where they have disclosed informatics-based approaches involving the starting from an RNA sequence and finding a chemical matter for that. So there are technologies like Informa, 2DCS, which is the combinatorial screening approach, as well as a few others, which actually can provide new starting points as well as understanding about the molecular recognition at the RNAs. Now, the use of virtual screening or the RNA structures has also been successfully tried. And this is actually another area where more structures of RNAs as well as folds will help us in taking this project forwards. We do have some experience with that. And I will sort of present one of the small work what we've done using the structure of RNA for virtual screening. And most of these another thing which I think will be very important in the RNA-based drug discovery, where the computational or the informatics-based tools can be very useful, is the use of the machine learning algorithms, because sometimes the SAR what we derive on the RNA target is very subtle, and sometimes it will be very difficult to actually make a trend analysis. And here I feel the use of artificial intelligence as well as machine learning algorithms can be very useful in deriving or driving the small molecule program. So as we move on, I think the development of screening technology is also very important, because I think that could be one of the important aspects of a small molecule drug discovery program. And we had some success with developing a fluorescence-based as a fresh-based technologies in RNA-target. More recently, actually, we also were able to use automated gene expression as well as quantification studies to actually run a small campaign in finding any hits. As you can see, we created a workflow which involves RNA isolation, cDNA synthesis, as well as treatment, as well as automated RT-qPCR to analyze the gene expressions, which is upon treatment with small molecules. And this was actually helpful in identifying some hits, which was then used as a starting point in the lead optimization campaign. At Syngene, I think we use different technologies or different capabilities to actually address some of the RNA-based small molecule programs. And I just wanted to sort of provide you some update in this and our experience on that. So more specifically here, the RNA-based library screening approach is another one which we use very often, wherein the common cores actually were used to derive in-house program, which then can be used to score these molecules and find drug-like starting points. And once these virtual compounds were then taken up for a library synthesis using our proprietary standard tools of library synthesis on solution as well as in solid phase synthesis. And we have actually also automated purifications, which actually helps in deriving a large number of compounds. In addition, we also were quite successful in deriving libraries where we have compounds with having ensemble of conformations, which actually helps in achieving the required goals for the RNA-based targets. Virtual screening is another approach, I think, where we had the limited success, I would say, for RNA-based targets, and I think primarily because of the complexity of the structure as well as the ensemble structures of RNA, making it more challenging to find small molecule hits. One of the biggest tasks in identifying small chemical probes is actually to find binding pockets, and use of chemical biology tools like photoaffinity labels has been quite successful, and we had quite success with use of azido as well as azurine chemistries to actually enrich the targets, and careful crafting of the biotin labels helped us to enrich the protein to about 3% to allow isolation as well as enrichment of the target and to move the SAR in this direction. So clearly there are several approaches one can use in identifying a chemical matter in RNA-based targets. Now, in one of the case, area where we had some, started working on is actually using computational or AI-based tools in targeting RNA target, wherein actually, as you can see that, we need to adopt to the RNA target using the ensemble structures as well as molecular dynamics structures as well as free energy calculations. So we did a compound screen of about 2,000 molecules from commercial sources, which was then expanded using the machine learning algorithms. Again, the hits were pretty low, and the molecules were very low active. So we were actually able to start off with a few molecules, which then progressed into a lead series. And currently, actually, these are being used for optimization. Again, we had a limited experience with this, showing the challenge with this particular RNA-based discovery. But I think as we advance into more machine learning algorithms, it will help us to optimize these molecules even further. So clearly there is a path forward for RNA-based small molecule discovery. Again, there are some new areas which I think will be very significant in targeting the RNA-based small molecules. And one of them is, of course, the RIBOTACs, which is a very new technology. I think is something that will play a major role in the coming days in terms of, basically here, these are ribonuclease targeting chimeras, where the work done again from the Matthew Disney's labs actually shows a pioneering expert in actually identifying a small molecule which can selectively or catalytically cleave pre-mRNA